Hi, everyone. I just want to thank Adeline and the organizers of GLIA for inviting us. As she says, sometimes the adult onset leukodystrophies are kind of overlooked, so I really appreciate the opportunity. My name is Quasar Padiath. I work at the University of Pittsburgh. Hopefully, I'll be able to give you an introduction and an overview of the Heatmap and some of the key successes and challenges of ADLD. I'll be followed by my colleagues, Dr. Raininko from Uppsala University and Julia Kofler also from the University of Pittsburgh, who will be talking to you about the clinical and radiological presentations and the neuropathology of ADLD. This is our work group membership. As you can see, it has quite an international flavor, and we're really excited that we had two members from the very recently formed patient advocacy group, the ADLD Center, who also joined us. And one of them, Bishan Singh, is actually in the audience here. So it's really great we have that advocacy group going on. Okay, so let's talk a little bit about ADLD. As Adeline was mentioning, so ADLD is a purely adult onset leukodystrophy. They're much rarer than the childhood onset leukodystrophies. And ADLD specifically has an onset of about the 5th to 6th decade of life. It was first described by Eldridge in 1984 and was diagnosed as a form of primary progressive MS. Unfortunately, it's a fatal disease. It's progressive. It has a median survival of about 18 years from the onset of symptoms. And it's an ultra rare disease. So we know for sure of about at least 30 families, and these are from all different parts of the world. Although the number of families might be relatively less, the size of the families are usually quite large. And that's because this disease presents much later in age. By that time, individuals have already had children. So if you have the mutation, you have a 50% chance of transmitting it to your child. So you have usually large families which have the disease phenotype. What are the symptoms? They usually begin with autonomic dysfunction, which present as either bladder, bowel, or erectile dysfunction in men, orthostatic hypotension, heat intolerance, followed by pyramidal symptoms, which include spasticity and weakness, cerebellar symptoms, which include ataxia and intentional tremor. Cognitive deficits have been reported, but these are usually much later in the disease. Both Raili and Julia are going to be talking about the radiological and neuropathological findings, so I'll just give a very brief one-line kind of description. In the case of ADLD, the MRI is characterized by symmetric T2-weighted hyperintensities in the cerebral white matter and an early involvement of the upper and middle cerebellar peduncles. The neuropathological findings are characterized by a vacuolar myelin loss with the preservation of oligodendrocytes and abnormal astrocytes. So the majority of ADLD cases are caused by duplications of this gene called lamin B1. A much smaller subset are caused by upstream deletions, and we'll talk about that in a little more detail in a few slides further down. Patients with the upstream deletions, many of them lack this earlier dysautonomia. So there's some degree of genotype kind of phenotype correlation. So what are the research challenges and strengths? So I think a major success is that we think we're pretty sure that the disease is caused by overexpression of this particular gene. We have a mouse model that recapitulates many, but not all of the features. It's not a perfect mouse model. But unfortunately, some of the mechanisms of demyelination are still unclear. We do not have good functional outcome measures, and disease burden has not been formally studied. But I think one advantage with ADLD is that patients seem to have a pretty stereotypical pattern of disease progression, so that might help kind of the course of action a little easier. There is no therapeutic target engagement, and we do not have response biomarkers, although I think Raili will show that the MRI could be used as a biomarker. And we do not have clinical trials at this time. Patient advocacy, I think, is a real strength, especially in the last few years, where we've had a patient community come together and they've actually started this advocacy group, the ADLD Center, and they've started a patient registry. So I think that's going to be really, really important for us as we move forward. Some of the clinical challenges and successes. So there's a really robust test for ADLD. It's a really simple real-time PCR test or an MLPA assay which can identify the duplication and the deletion. And it's kind of paradoxical, but some of the more advanced and the fancier next-generation sequencing and the exome panels can actually miss this duplication or deletion. And I've had some cases from GeneDx where they could not identify the duplication. So that's something to keep in mind. And, as we've heard, genetic testing can be a barrier for international families. There is no clear pathway for newborn screening, and we have not yet identified any disease-modifying therapies. And I think diagnostic awareness will be really important, hopefully at some point of time when we identify a therapy for the disease. I think the major issue is that because ADLD is such an ultra-rare disease, you have individual patients be seen by doctors all over the world. So there is no real standardized care. And I think as we were discussing in the patient group, we thought that having a standardized set of care was really critical for the ADLD community. So I'm hoping that this is something that can grow out of our work group meeting. So let's talk. So this is the heat map, and I was so envious when I saw the previous group's heat map, which is all blue and green. I don't think I've ever seen blue. So, unfortunately, we're not there. We have a lot of reds and oranges, but I think we do have strengths in the patient advocacy and some of the preclinical disease models and understanding the disease mechanisms for this disease. Okay, so let's talk a little bit more about the disease mechanism, which I think or the neuropathology, which I think is kind of one of the successes for ADLD. Okay. So like I said, ADLD is not kind of your typical mutation. It's not like a mutation in the gene or a change in amino acid. It's actually caused by what is known as a genomic duplication. So this entire genomic region, which contains the lamin B1 gene, is duplicated and inserted next to the original copy. So it's a tandem, what we call a head-to-tail duplication. So patients will have 3 copies of the gene, and normal individuals will have 2 copies of the gene. And we think as a result of this, there's an overexpression of the protein and the RNA, and we've tested this in many patient tissues. But of course, things are never as simple as it is. So we think that it's a much more complex mechanism, which is targeting overexpression. And unfortunately, I don't have time to go into that at this meeting. So every family has a unique duplication. So duplication sizes vary in different families. But if you notice, all of them have duplications which encompass the lamin B1 gene. And the vast majority, pretty much all of them, except for one case, are actually this tandem head-to-tail orientation. Once we first identified the duplication, and subsequently Alfredo Brusco's group identified a family with a deletion upstream of the lamin B1 gene, and that's shown here, and we've identified 3 or 4 more families after that. In both cases, we think that both the duplication and the upstream deletion result in an overexpression or more amount of this lamin B1 protein, and that's what we think is causing the disease. So what is lamin B1? I've been using that term for a long time. What exactly is lamin B1? So lamin B1 is a component of the structure known as the nuclear lamina. If you look at the schematic of the nucleus here, you see this thin pink ribbon-like structure. That is the nuclear lamina. And it's shown in the STORM image here. We actually show that lamin B1 is in the outermost layer of the nuclear lamina. So lamin B1 is an intermediate filament protein. And what it does is 2 dimers wrap around each other and produce these filaments. And these filaments produce a meshwork which is just underneath the nuclear envelope. And it's thought that this meshwork provides structural integrity to the nucleus. So if you remove that, the nucleus gets all floppy and deformed. But apart from that, it also has very, very important roles in chromatin organization and gene expression, DNA repair and replication. And lamin B1 specifically has been shown to have very, very important roles in cell proliferation, senescence, and aging. So for us, when we first identified the gene, it was a really fascinating question, because you now have a protein that's pretty much expressed in all cells, right? Why on God's earth would it specifically cause a demyelination phenotype? There was absolutely no direct connection between this subcellular component and myelin, right? So what cell type is affected? Is it oligodendrocytes, astrocytes, neurons? Oligodendrocytes are an obvious kind of culprit because maybe they're the ones that produce myelin, right? But it could also be astrocytes because they signal to oligodendrocytes, and maybe that's what's defective. Or maybe all three are involved, so we really don't know. So some of the questions, and of course, why is the phenotype age dependent? Why is it that individuals seem to be perfectly normal till they're about 30 or 40, and after that they start exhibiting demyelination? So these are all really fascinating questions, and we've been trying to answer some of them using some of our preclinical mouse models. So this is a model that we made a few years ago where we introduced the human lamin B1 gene under the control of a PLP promoter. So PLP1 is a gene that's highly expressed in oligodendrocytes. So this promoter targets the expression of lamin B1 specifically to oligodendrocytes. And because it has a flag tag, you can actually distinguish the exogenous and the endogenous lamin B1. So you can see there's probably about twice the amount of lamin B1 as compared to the wild type. Right. So when we made these mice looked perfectly fine when they were born. You could not distinguish them from wild-type mice, but as they progress, you could see a progressive kind of worsening of the muscle phenotype, and you had significant weakness, and the mice looked really horrible, as you can see, by the age of about a year. And they had significantly reduced survival. These mice also showed a very characteristic, the characteristic vacuolar demyelination. This is in the spinal cord. This is a myelin stain. And on EM, you can see a lot of these vacuoles. So to some extent, to a large extent, I think we're quite happy with this mouse model because it recapitulates many of the features. Of note, this mouse model does not exhibit any autonomic dysfunction. So I think that's something that's quite interesting, and we don't know exactly why that is. We had done some biochemical analysis, or we had done some RNA-seq analysis on these mice, and one of the pathways that popped up was this pathway of cholesterol biosynthesis. And again, I won't go into all the details. It's published, and we've done a lot more work after that, but one of the pathways we think could be playing a role is that you have an increased level of lamin B1, you have decreased expression of these myelin genes, resulting in a decreased expression of myelin-rich lipids, which lead to a demyelination phenotype and an inflammation phenotype. These seem to kind of play off each other, ultimately leading to the ADLD phenotype. We have some clues about cell type specificity. So we made a similar mouse, kind of a similar mouse where we overexpressed lamin B1 in astrocytes, but they did not appear to have a demyelination phenotype. Interestingly, those mice do show nuclear abnormalities, but they do not have kind of a demyelination phenotype. So, at least for the mouse model, it suggests that maybe oligodendrocytes are the driver, but, it could also be that there are multiple different roles that are played by different cell types. So let's talk about treatment. So I think the one advantage with ADLD is that it's really ideally suited for early therapeutic intervention. And the reason for that is if you have ADLD, it's very, very likely, at least all the years I've worked with this disease, I've never come across a de novo case. There's always been some family history. So if you have ADLD, there's a very strong chance that you have one of your parents have had the disease. And because there's robust genetic testing, you can get tested very early. So you have a huge therapeutic window of nearly 30 or 40 years within which you can start therapy. So I think that's really, really important. And one thing that we are all kind of, I think, agreed upon is that it seems like this overexpression of lamin B1 seems to really be what's causing the disease. So most of our kind of approaches for therapy have been in trying to reduce the levels of this particular protein. We can do this very easily in vitro. So if you overexpress lamin B1 in vitro, you can get all these really weird-looking nuclei, and if you knock it down, you can rescue that. But what we really wanted to ask is reduction of lamin B1 in vivo a viable therapeutic strategy, right? Because that's a really, really important question. Because if it is not, then you really don't want to waste your time developing all these different kinds of tools to reduce the level of this protein, right? So this is a really critical question for us to answer. So what we did was we used the similar mice, but what we then did was made in other mouse model where we introduce LOXP sites on either side of the lamin B1 construct. And the advantage with LOXP sites, so first of all, these mice are very similar to our original transgenic mice. They also express the overexpress to the similar level. They target the overexpression specifically to oligodendrocytes, and these mice also show an age-dependent motor dysfunction and have significantly reduced survival. The advantage with these mice is that you can now cross them to inducible Cre mice. And if you inject these bitransgenic mice with tamoxifen, you can actually excise out the overexpression cassette. Right? And we can do that quite well. You can see that when we inject tamoxifen in these mice, this overexpression cassette, which is about 2 kb, is completely cut out. And we can also reduce the overexpression of lamin B1 both at the RNA and protein level quite significantly. So we thought that these would be, this is a relative, obviously this is not translatable to humans. Unfortunately, humans don't have LOXP sites on either side of the lamin B1 gene. But you can use this to understand whether removing or reducing lamin B1 levels will be useful. So we had two treatment paradigms. One was the pre-treatment paradigm where we inject tamoxifen before the onset of symptoms, and one is the reversibility paradigm where we inject tamoxifen after the onset of symptoms. Again, these mice have been very, very recently made, so we've just started working on the pre-treatment paradigm, but there the kind of results look interesting, and the plan is to use behavioral, survival, and histopathological assays to see how it affects disease progression. So this experiment obviously helps us answer the important question of whether reducing lamin B1 in vivo can ameliorate the disease, but it also helps us identify a therapeutic window, right? Is it that you have to reduce it before the onset of symptoms, or can you also reduce it after the onset of symptoms? And that's very, very important for patients, right? And also answers a mechanistic question. Do you require this elevated level of lamin B1 to continuously be present for the disease, or if you remove it, can you prevent kind of the disease progression? So we use two paradigms. So this is a rotarod paradigm, and here you can see all these animals are injected with tamoxifen. So these are the animals which are the TG flox PLP Cre ER, where you are excising out the overexpression when you inject with tamoxifen, and these are the animals which are just TG flox, which do not have a removal of the overexpression. And you can see, so we inject tamoxifen in this case at about 3 months here. You can see that they start out nearly the same, but it's pretty obvious that the animals which are injected with tamoxifen and have the overexpression removed have a much, much better kind of outcome. So it looks like, at least behaviorally, they look much better. We also perform what we call a functional observation battery, which is just different phenotypic parameters, like movement and the condition of their fur. And you can see that the TG flox animals become much, much worse over time, whereas there's only a very, very slight deterioration in the animals where we reduce the overexpression. So at least from this work, it seems like that the pre-treatment paradigm seems to be really effective. We're going to try the reversibility paradigm also. So this is kind of at least a bit hopeful. So these are just the videos of the mice. So this is, I think these are mice at about 9 or 10 months old. So this is a wild-type mouse. This is a TG flox mouse. You can see that it barely moves. It's highly ataxic when it moves. And this is a mouse where we have excised out the overexpression cassette at about 3 months, and for all practical purposes, it's kind of indistinguishable from the wild-type mice. Okay. So just to kind of recap and kind of summarize the work, from the work group, we thought that, and what's really critical is the implementation of some kind of uniform criteria from the diagnosis and disease progression, and that really helps us kind of normalize and standardize work across different clinical centers. Because like I said, we have individual patients all over the world. It's impossible to get everybody in one center, so I think this is really critical. Because this is an adult onset disease and has such a long course of disease, it's very tough to do a clinical trial. So it's really critical to identify biomarkers that can serve as surrogates for disease progression and response to therapy. I think we have some promising leads in looking at therapeutic targets, especially for lamin B1. We are working with Ionis Pharmaceuticals to develop antisense oligonucleotides to downregulate lamin B1 expression. We have also conducted a large small molecule screen for small molecule compounds that can reduce lamin B1. We have some really promising targets that we want to now try in vivo. Again, Alfredo Brusco's group had done a similar piece of work using FDA compounds, and then they identified alvespimicin, which I think is an HSP90 inhibitor, but I think they have issues with kind of toxicity there. We are also looking for AAV-mediated RNAi therapy and CRISPR genome editing to see if we can downregulate the overexpression. So I will stop there. I just want to acknowledge my lab members. Obviously, this is all before COVID. Bruce and Guillermo did a lot of the work. All our collaborators, both at Pitt and Italy, and clinicians around the world, our funding sources. A real shout out to the ADLD Center because they've been so supportive in the last few years. And most importantly, to the ADLD patients and family members, it's a huge, huge thank you. I've known these patients for many, many, many years now, and I think the fact that they have a fatal disease really gives us a lot of urgency to our work. So I'll thank you there, and then hopefully Raili can come and start talking. Thank you.
So, hello everybody. I will continue by talking about the radiological and clinical features of ADLD. And I prefer to use that name LMNB1 related ADLD because there are many other leukodystrophies which also have autosomal dominant heredity. These are typical MR images of an ADLD patients with relatively mild symptoms. And can you move closer to the mic, please? Is it better now? Okay. Quite large hyperintensities in the cerebrum. And hyperintense pyramidal tract, you can see here, here, and here. So it is the whole tract which is sick. And the typical finding is involvement of cerebellar peduncles. There are some few diseases with the same radiological finding, but they are clinically totally different. So it is not a differential diagnosis problem. And the most characteristic finding we can see here, around the lateral ventricles, there is a rim in which the white substance is less affected than, thank you, thank you, less affected as in other parts. And we can see that in T2 weighted images. Always when we talk about MR findings, we must remember that they always are sequence dependent. And 1 weighted images usually are produced with the FLAIR or SPINECO or SE sequences. And here we have T2 weighted SPINECO image and this typical rim around the ventricles. And that finding we can see in every ADLD patient with that sequence. But if we use FLAIR, we can have the same finding as in the earlier image. But look at this example. Here, the highest signal intensity is here around the ventricles. So the findings in FLAIR imaging are varying, and therefore I recommend to use both SPINECO and FLAIR sequences in patients which have ADLD or ADLD has been suspected. We make a study in Swedish patients. They were from 2 families, and we recruited real patients with symptoms, but also asymptomatic family members. And after genetic tests, we excluded those who did not have LMNB1 duplication. Thereafter, we had 23 subjects, and they all had pathological changes in MR images. And there was 4 subjects which were asymptomatic. And this asymptomatic period was almost 10 years even in this case, and here it is about 15 years. And then 3 of them developed a clinical disease, and the 4th one died in the car accident. In those asymptomatic patients, we can see how this disease developed. The first changes were always seen just under motor cortex in the upper part of the pyramidal tracts. Then they spread downwards along the tracts through the internal capsule, cerebral peduncles, the pons, and medulla oblongata. After that, the cerebellar peduncles become hyperintense, as you can see here, and at the same time, cerebral white matter changes grows. And first involved are frontal and parietal lobes, then the involvement spreads into occipital lobes, and the temporal lobes are the last affected. We created a grading system, grades 1 to 5, to use these findings for comparisons with the symptoms. Here we can see examples of 2 patients. The first one had grades 1 to 3, and the other one from grade 3 to 5. And here we can see those grade 3 changes both in the flare sequence and in the spinnacle sequence. In the left graph, you can see how the symptoms develop. This yellow area is the period during which the patients got clinical onset. Then we used the Kurzges's Expanding Disability Status Score, EDSS, and what can we see? That disability, of course, starts a little later, and then the state become worse in the same way than in MRI images. We also examined the spinal cords in 14 subjects. In 9 of them, we had a follow-up from 2 to 10 years. And in the image on the left, you can see a transverse slice at C2 level from a healthy control. And these 2 images are from the patients. If you look at the spinal cord, it is much thinner in patients, and in follow-up studies, we can see that atrophy was progressive. All subjects, even asymptomatic ones, had high signal intensity changes in white matter in the spinal cord. Here in the healthy control, the signal intensity is almost homogeneous, but in the patients, we can see white matter as a hyperintensive structure. And in one asymptomatic patient, we saw changes locally in cervical area, but in all other cases, whole the spinal cord was involved. What successes have we had? It is success that we can get a specific diagnosis using MR findings of the brain with the combined with the clinical symptoms and history of autonomic symptoms. And diagnosis can be obtained even in asymptomatic subjects about 10 years before clinical onset. Early spinal cord involvement has also been shown with MRI, and it may explain autonomic symptoms, which usually are the first symptoms, and also a part of motor symptoms like spasticity of the legs. MRI can be used as a biomarker if any therapy will be available, and even under therapy development, it can be used even in animals. And challenges. Great majority of the patients have the clinical and radiological findings such as I have shown. But there are some variations. There are some patients who doesn't develop automatic symptoms or develop them quite late. There are patients which have no changes in the spinal cord and very mild or no changes in the brainstem or cerebellum. And those variations can be related to the type of the genetic defect. Some of them had duplication, some may have deletion, but we have very few cases, so this is a branch in which we need more research. Thank you.
Okay, I will wrap up this session by presenting some of the neuropathologic features. I'm a neuropathologist at the University of Pittsburgh, and I had the opportunity to study 2 autopsy brains of 2 female siblings that both passed away in their early 60s. And it shows some of the characteristic findings that can also be seen on MRI images, and that is this pretty widespread demyelination in the centrum semiovale, with the very disease-specific sparing of the deep periventricular white matter. Both frontal, parietal, and occipital lobes are usually more severely affected than the temporal lobes. On higher power, you can see that these areas of demyelination are somewhat patchy in nature, what's sometimes described as this more tigroid pattern. And what you can't see from the images, but what you can see when you actually handle those brains, that these areas of demyelination are very soft. So they don't have these features of sclerosis that you typically see in areas of scarring and demyelination. And as seen on the MRI images, other areas affected are the cerebellar white matter and to varying degrees the corticospinal tract, which were actually rather mildly affected in our patients. This relates to the classical findings on microscopy here, Luxel fast myelin stain, that again shows these areas of severe but patchy demyelination in the upper white matter with relatively sparing of the deep periventricular white matter region. And again, on higher power, you can see this really rarefaction of the white matter with occurrences of these vacuoles. It's true, a true demyelinating disease in the sense that while we do have axonal loss, there's definitely more myelin loss than axon loss. As you can see, there are some naked axons that have lost the myelin sheath, but eventually axons appear to die as well. What we don't see is the loss of oligodendrocytes. So it's not a disease of dying of oligodendrocytes. We haven't perform formal quantifications, but at least visual impression is that there are similar densities in affected and non-affected white matter areas. We do get some microglial activation. You can see here some activated macrophages, but it's not as dense and inflammatory infiltrate as in many other leukodystrophies. But what's most remarkable is that there is an absence of reactive gliosis in these affected white matter region. Again, here on the low power image of a frontal lobe section, you can see a normal complement of astrocytes in the overlying cortex. There's still some astrocytes left in the deep white matter, but the area of demyelination, you really have a loss of astrocytes and no reactive gliosis, and that explains the soft consistency that we see on gross examination. And not only do we lose the astrocytes, the ones that are left behind here on the lower panels are highly abnormal in their morphology. They are fragmented, they have abnormal morphologies, and you don't see this diffuse gliosis as, for example, here in the upper right corner in a Krabbe disease brain. And even in the areas where on low power you still saw some astrocyte staining, even in those areas, the astrocytes have clearly abnormal morphological features. So in order to dive a little deeper into what's going on in ADLD, we started doing some lamin B1 immunohistochemistry. And as Quasar has mentioned in his introduction, lamin B1 definitely decreases as we get older and is actually often used as a senescent cell marker. So you can see here on the top, and these are really just some convenient samples, we have really strong expression in the gray mat of Krabbe disease brains, but in 80-plus-year-old Alzheimer's disease brains, these are for multiple different cases, we clearly see a loss in nuclear staining intensity. And this is different in our ADLD cases, at least in the cortex, we really see strong maintained lamin B1 expression, at least until their 60s. We haven't really done a formal study comparing age-appropriate controls, so this is something we need to do. But it definitely seems to be some preservation, high expression of lamin B1. And the lamin B1 expression, interestingly, is very cell type dependent. So if you use some co-labeling with different cell lineage markers, we can see that really the strongest labeling cells are the microglia and macrophages, but you can see in the background here in the affected white matter all the numerous persistent oligodendrocytes that still have a pretty decent lamin B1 expression. But when we look specifically at astrocytes, we really are unable to detect any significant nuclear lamin B1 staining. There seems, again, some really atypical forms, and none of them really has any residual lamin B1 expression. So that really leaves the question is this really the end stage of the disease where lamin B1 has downregulated? We really don't know, and it's hard to draw causal inclusions from a pathology image, but it definitely raises a lot of questions what's going on with lamin B1 and what the affected cell type is. And just for a contrast, other diseases do have also decreased astrocyte expression, but it's still persistent. So here in a Krabbe disease brain, all those reactive astrocytes have still persistent lamin B1 expression, and the same here in our Alzheimer's disease brains. So there have been some questions raised about the roles of astrocytes in ADLD pathogenesis. So in our human brains, and this has been reported before, there's clearly astrocyte loss. We lack the typical fibrillary gliosis, and we have abnormal astrocyte morphologies, which clearly raises the question if where in the pathogenesis astrocytes involved. We also see a decrease in lamin B1 expression. Is this just an indication that they are undergoing some premature senescence, or is this something that's really driving the disease? Is something we don't know yet. Next step is to do some senescent markers on these astrocytes to see what phenotype they have. In the mouse, as Quasar has mentioned, if you overexpress lamin B1 in astrocyte-specific models, it does not produce a disease phenotype. So it doesn't seem to be that astrocytes alone would be able to drive the disease process. So possibly it's an interaction between the different glial cell types. Other studies have overexpressed lamin B1 in astrocytic tumor cell lines, the glioblastoma cell, and clearly see some abnormalities there. But the question is if this can be really translated into the more milder expression that we see in our ADLD cases. So to summarize, what we see in ADLD as a neuropathologist is this vacuolar myelin loss in characteristic distribution of the brain affecting the centrum, Samuel Wallace, cerebellar white matter, intervarying degraded corticospinal tracts with relative sparing of the periventricular white matter, which is one of the more characteristic features of the disease. Which I haven't shown here is we really see largely preservation of neuronal cell population. We see preservation of oligodendrocytes, but we have this really abnormal astrocyte phenotype. So challenges that we have from a pathologist perspective, or what we can learn from studying human autopsy brains, is that there's clearly a role for astrocytes in the disease pathogenesis, but we really don't know where in the causal step these astrocyte pathologies fall into. So are they really driving the disease, or are they sort of like a bystander of the abnormal oligodendrocyte phenotype and some abnormal interactions between astrocytes and oligodendrocytes? So right now, Quasar is planning to do some single-cell RNA sequencing studies using our human autopsy material. We're hoping that it may help us elucidate some of these glial phenotypes. But we also don't know, both of our brains came from patients with the standard lamin B1 duplication. As far as I know, nobody has done any autopsies on a deletion case, so we don't know if we have similar pathology features as in the standard mutation. And there's I'm probably biased as a neuropathology. I think we can learn a lot of human postmortem brain tissue samples for all of our leukodystrophies, but for many of the diseases, there's really a shortage of human autopsy brain samples to study. So if anybody is interested in reaching out, I'm always welcome to increase brain donation or help with brain donation programs. So with that, I would like to wrap up and with the 5 minutes.
You did a good job. If we could have the speakers come, we'll maybe take a couple questions.
Hi, great presentation. I have a question regarding the neuropathology and a comment, because I'm a neuropathologist too. And the question is, what do you think is the cause of the patchy myelin involvement?
There has been some discussion in the literature that it's related to perivascular preservation.
Like in PMD?
I haven't really seen that consistently in ADLD brains. Of course, you always have to take into consideration what I see under the microscope is a two-dimensional picture. I never know where in the 3D plane the next vessel is. So, but it's not a really very obvious perivascular preservation pattern. So I really don't have a good answer to your question yet, what the patchy nature is.
That's why I was asking. And then I have a comment. I had the opportunity to examine the brains of the Italian cohort of ADLD patients, including patients with deletions. So I have found exactly what you found in your patients, and in also the same pathology in the patients with deletions, with two things. One, that the astrocytes are immature, and I suspected that they're unable to make reactive gliosis because they are immature, and this has been confirmed by people in Italy also on iPSCs-derived astrocytes. And two, that the patients with deletions often have a relative sparing of the infratentorial structures, and there the astrocytes are much more normal.
All right.
Hi, a question for Quasar. It was a great presentation. I just want to point out, did you see any similar pathology in the mouse, in the supratentorial region, in the CNS brain?
So I have to qualify that the mouse is a pretty artificial system. So we use this PLP promoter, and then when we looked at the way the PLP promoter expresses, it seems to express to the highest extent in the spinal cord. So I didn't show that because I didn't have time. We really saw the maximal demyelination in the spinal cord, and that's why I showed spinal cord for my most of my slides. So, it does not recapitulate perfectly the anatomical distribution of the demyelination. So I don't think it would make sense to look at those structures. So we looked, the spinal cord really had the major amount of demyelination, and then we could see a little bit in the cerebellum, but there was not that much, affecting the cerebral white matter. And I feel that's not really a consequence of the pathology per se, but more an artifact of the way the overexpression is distributed. There's much more overexpression in the spinal cord compared to some of the other features, other areas.
And just a follow-up question, the laminin is very, makes a complex with B1, laminin B1, C and A. Since you did a transgenic mouse, do you think they're gonna make a complex, the human with the mouse the same way?
So I think both the human and the mouse lamin, I think, are about 98% identical. And we did confirm, so because the mouse has the human lamin has a flag tag, we were able to show that they're perfectly co-localized. And, people use human and mouse lamin interchangeably. So I think they would interact in a very, very similar fashion.
Thank you.
Go ahead.
Sure. As a pathologist, have you seen extra neurological signs of involvement, particularly liver and heart?
Both of our cases were restricted to the brain and spinal cord, the autopsy, so we haven't had the opportunity to do any other organic examinations.
If I can ask a question, it sounds like your group has made and your disease group has made such significant progress in understanding the pathophysiology and thinking about potential therapeutic strategies through downregulation. From the perspective of clinical trial readiness, have you thought about sort of potential outcome measures that, beyond MRI biomarkers, that would be things that you would sort of start getting ready in parallel so that once your trial options are ready, you have a ready trial design?
So I think that's absolutely true, and we have not thought about that. And I think having this work group come together, I think that's one of the major gaps that we discussed, and I think that's something that we should definitely be addressing. And I think in terms of biomarkers, I think MRI is definitely one of them, but we're also looking at other biochemical markers as surrogates. So that's something that we are looking at.
Okay.
Thank you. So based on the senescence thing, that's what I was like, I've heard of this gene before. So can you guys comment on the senescence? And also, I know it's ultra rare disease, but is there anything known about cancer predisposition in this population?
The senescence, it's really fascinating. So in the last few years, the downregulation of lamin B1 expression is one of the most robust markers for senescent cells. Right? So we know that if cells undergo senescence, they do have a downregulation of lamin B1. Whether that drives senescence or not is a very different question, and I think there is no clear answer to that. And again, I didn't have time to show a lot of the other data, but we do have some really interesting data. So I'd shown that lipid downregulation is one of the pathways. And if you actually look at senescent cells, one of the pathways that's a critical part of senescence changes is a downregulation of lipid biosynthesis. So we think what's happening is that the upregulation of lamin B1 is somehow triggering senescence-associated pathways, even in cells like oligodendrocytes, which are terminally differentiated, right? And they're not really going to proliferate anymore. So that's the first part. And there have been reports of lamin B1 changes in cancers. I don't think we've not come across any patient any history that suggests that these patients have any difference in their predisposition to cancers. But I think some pancreatic cancers supposedly have shown increase in lamin B1 levels. But I do want to caution that lamin levels are so very sensitive to cell type that if you take a bulk of cells, you really don't know what kind of cell types there are, and different cell types have very, very different levels. And it's also uniquely sensitive to things like the density of growth and the rate of proliferation. So I would be very cautious about making a direct correlation between lamin levels and other diseases.
All right. And on that note, we're going to take a brief break. I'm going to ask everybody to be back here at 2:55.